Okay, welcome to the Chapter 12 notes. Let's go ahead and get started. You can see Chapter 12 is broken up into 4 different sections, and you see the different titles there. So for Section 1, the vocab double helix and nucleosome. First up, a little history. So these different scientists, they came at different times and did some pretty important work for what we know now. So we'll get into them. So this scientist Griffith, performed the first major experiment that led to the discovery of DNA as the genetic material. And you can see what's going on here. You guys read about this. This was within your chapter 12.1 intro. Another scientist, Avery, identified the molecule that transformed the R strain of bacteria into the S strain. Concluded that when the S cells were killed, DNA was released. The R bacteria incorporated this DNA into their cells and changed into S cells. Hershey and Chase used radioactive labeling to trace the DNA and protein. Concluded that the viral DNA was injected into the cell and provided the genetic information needed to produce new viruses. You can go ahead and do this on your own. I can't move these for whatever reason. So you can take a look at those, pause it if you'd like. Moving on, DNA structure. Nucleotides. Nucleotides consist of a 5-carbon sugar, which is right here, a phosphate group, and a nitrogenous base. And this nitrogenous base is what changes within DNA structures. So this base can be adenine, guanine, cytosine, thymine, or if we're dealing with RNA, it can be uracil instead of thymine. I'll get more into that later. The sugar pertaining to DNA, the sugar and phosphate will remain the same. So there's this rule, Chargaff's rule, where C and G will pair with each other, T and A will pair with each other. So in theory, however many C's there are, cytosines, there should be the same number of G's, guanines. So that would represent C equaling G. And you can see in these different species, these different organisms, that's pretty close to true. Of course, things can happen, exceptions, and then the same with T and A. Now we're dealing with DNA here. That's why there's the T. So the thymines and the adenines should be the same or very, very close to the same. So you see the percentages based on the different bases within the DNA structure, and they are the A's and the T's are significantly close, and the G's and the C's are significantly close. X-ray diffraction. X-ray diffraction data helped solve the structure of DNA. Indicated that DNA was a double helix, and that's pertaining to the actual physical shape of it. Watson and Crick, they built a model of the double helix that conformed to the other's research. So a few things here. 2 outside strands consist of alternating deoxyribose, and that's the sugar, and phosphate group. Number 2, cytosine and guanine base pairs to each other. Cytosine and guanine bases pair to each other by 3 hydrogen bonds. Thymine and adenine, however, those bases pair to each other by 2 hydrogen bonds. Continuing with the DNA structure. DNA often is compared to a twisted ladder. The rails of the ladder, or the sides, if you will, the vertical parts, are represented by the alternating deoxyribose sugar and phosphate group. The pairs of bases, whether it's the C or G, C and G, or T and A, they form the steps of the ladder. So here we see, just going back to that ladder analogy, this would be, think of like a ladder on its side laying on the ground, right? So here are the sides of the ladder, the sugar, phosphate, and here are the steps of the ladder. Obviously, if you were using the ladder, it would be up and down, okay? So orientation, on the top rail, the strand is said to be oriented 5' to 3'. The strand on the bottom runs the opposite direction, and is oriented 3' to 5'. So, starting with DNA, kind of just going over the basics of the DNA structure, which then leads us to chromosome structure. DNA coils around histones, which are proteins, to form nucleosomes, which then coil to form chromatin fibers. The chromatin fibers supercoil to form chromosomes that are visible in the metaphase stage of mitosis. And that's it for section 1. Section 2 vocab, semi-conservative replication, DNA polymerase, and Okazaki fragments. First up, semi-conservative replication. Parental strands of DNA separate, serve as templates, and produce DNA molecules that have one strand of parental DNA and one strand of new DNA. Unwinding. DNA. Let me do this, probably be easier. There we go. Let me know if that's better. Unwinding, sorry about that. DNA helicase, an enzyme, is responsible for unwinding and unzipping the double helix. RNA primase adds a short segment of RNA called an RNA primer on each DNA strand. Base pairing. DNA polymerase continues adding appropriate nucleotides to the chain by adding to the 3' end of the new DNA strand. You can break this down, this image here. There's a lot going on here within this image, and this was part of, I believe, if it wasn't on the intro, it was definitely on the study guide. Continuing on with replication. One strand is called the leading strand and is elongated as the DNA unwinds. The other strand of DNA, called the lagging strand, elongates away from the replication fork. The lagging strand is then synthesized discontinuously into small segments. Those small segments are called Okazaki fragments. Joining. DNA polymerase removes the RNA primer and fills in the place with DNA nucleotides. DNA ligase then links the two sections. Comparing DNA replication in eukaryotes and prokaryotes. Eukaryotic DNA unwinds in multiple areas as DNA is replicated. However, in prokaryotes, the circular DNA strand is opened at one origin of replication. And that's it for section 2. Section 3, some vocab here: RNA, messenger RNA, ribosomal RNA, transfer RNA, transcription, RNA polymerase, codon, intron, exon, and translation. So the central dogma. This was a question on one of the intros. Must have been section 3, obviously. It was either on the section 3 intro or the study guide dealing with this. So RNA contains the sugar ribose and the base uracil instead of thymine, as I mentioned, I believe, in section 1. So in RNA, the sugar is now a ribose sugar, not a deoxyribose sugar, which is in DNA. And that's why DNA is called DNA, because DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid because it has the ribose sugar. Anyway, getting back to this, sorry about that. RNA is usually single-stranded, and it's very important that it's single-stranded, and I'll show you why towards the end of these notes. Okay, 3 different types we're going to look at. First up, messenger RNA, also called mRNA. Messenger RNA are long strands of RNA nucleotides that are formed complementary to one strand of DNA. The next type, ribosomal RNA, also known as rRNA, associates with proteins to form ribosomes in the cytoplasm. And then lastly, the transfer RNA, also known as tRNA, are smaller segments of RNA nucleotides that transport amino acids to the ribosomes. So I'm hoping, as you're hearing me say these different things that we've gone over in previous chapters, that you're at least remembering what they are. Things like amino acids, things like ribosomes. So here is a chart to go over the function of the different RNAs, and you can go ahead and pause it and try those out. All right, first up, transcription. Through transcription, the DNA code is transferred to mRNA, messenger RNA. This is happening in the nucleus. The DNA is unzipped in the nucleus, and RNA polymerase binds to a specific section where an mRNA strand will be synthesized. And you can see that happening right here. Here's the RNA polymerase, and then you see this mRNA strand being transcribed. And notice the A's pair with U's. The C's and the G's still pair with each other. The T's still pair with A's. However, the A's pair with U's. So that's one thing I want you to be aware of. RNA processing. The code on the DNA is interrupted periodically by sequences that are not in the final mRNA. Intervening sequences are called introns. Remaining pieces of DNA that serve as the coding sequences are called exons. The code. Experiments during the 1960s demonstrated that the DNA code was a 3-base code. The 3-base code in DNA or mRNA is called a codon. Okay. Now, dealing with translation. This is known as a codon chart. And when we're looking at the mRNA sequence, they are packaged together in 3 bases. We'll get to that in 1 minute. So, translation, here we go. In translation, tRNA molecules act as the interpreters of the mRNA codon sequence. At the middle of the folded strand, there is a 3-base coding sequence called the anticodon. Each anticodon is complementary to a codon on the mRNA. So on the mRNA, you just have the codon. Let's say it's AUG. So the AUG mRNA codon, we look that up here on the codon chart. A is the first base. A, let's look up the second base, U. So we have AU, so we're in these 4. Obviously, you see where it is. And then G on the third. So it's right here, AUG. That's known as the start codon. However, if an AUG is within a translating sequence, then it will translate to a methionine amino acid. So these different names, and as you can see, some code for the same amino acid. I just gave away the answer. So all these different names, these are just the different amino acids. And remember, the goal here is we're to synthesize proteins. This whole process is known as protein synthesis. And in order to synthesize a protein, we need to put together amino acids. And that's what's going on right here. Okay? So we're essentially translating, that's why it's called translation. We're translating the mRNA sequence, which we got from the DNA, into an amino acid sequence. And once the amino acids are put together, then the protein is then synthesized and basically shipped out to wherever it is needed. All right. Okay. So here is a nice little diagram. I know it looks like there's a lot going on and your brain's probably going crazy and you're saying, well, we're not in school, so yada, yada, yada. So let's just try to break this down. So we'll start up here. Okay. Here, all right, before we even start, let's give perspective here. We are looking, we're inside of a cell right now. Think of just an animal cell, if you will. Here we have the nucleus, this purplish structure, if you will. We have the nucleus. Here's a nuclear pore. Okay. So the DNA is in the nucleus. And then you have the mRNA being transcribed from the DNA template by this RNA polymerase. Okay. So going from DNA to RNA, that is transcribed mRNA. That's known as transcription. Okay. Then all these little loops and ribbon, if you will, think of it as ribbon. Okay. Now, the thing I want to point out is that the RNA is single stranded as opposed to the double strand of DNA. So here we have the RNA processing before. Whoop, what happened there? Okay. The RNA processing introns and are excised, and the mRNA is processed. Okay. So anyway, we get to the nuclear pore where the mRNA is then released from the nucleus. Now it's outside the nucleus within the cell. Now, DNA, it's double-stranded, so it's typically too large to go out of the nucleus. However, RNA, single-stranded, think of it as being smaller in a sense, it can then leave the nucleus. All right. So the mRNA leaves the nucleus and associates with the ribosomal subunits. Okay. So it meets up with these subunits, and eventually we get amino acids, and this is where the whole we'll get eventually to the translation process. Amino acid activation. An enzyme activates tRNA by attaching a specific amino acid to each tRNA. And these are known as those anticodons, as you see here. But we are worried about the codon. Okay. So here we have the RNA, and translation occurs to essentially create the start of a protein. tRNAs add their amino acids to the polypeptide chain as the mRNA moves through the ribosome, one codon at a time. When a stop codon is reached, the translation terminates and the polypeptide is released. So that's what those, if I go back, that's what those stop codons are. Basically telling this ribosome, okay, stop putting these amino acids together, and we'll wait for another strand of mRNA to be put out from the nucleus to then be translated into amino acids and so on. All right, I know that's a lot. Try to listen to that over and over again, but I think this picture does a pretty good job. I know there's a lot going on here, but just follow along, stay up here, just go from A to B to C to D to E. All right. Okay. Moving on. One gene, one enzyme. The Beadle and Tatum experiment showed that one gene codes for one enzyme. We now know that one gene codes for one polypeptide. All right. That's just a little extra. Give them some credit. Section 4, gene regulation, operon, mutation, mutagen. Prokaryote gene regulation. The ability of an organism to control which genes are transcribed in response to the environment. An operon is a section of DNA that contains the genes for the proteins needed for a specific metabolic pathway. Operator, promoter, regulatory gene, genes coding for proteins. So let's break it down. The TRP operon. This is because of TRP because of tryptophan, which is an amino acid. All right. And we're not going to go into too much depth of these. Okay. Same thing with the lac operon. If you want to dig into these further, go for it. But not going to be focusing too much on this stuff right now based on our situation. Okay. All right, good. Let's see. Eukaryote gene regulation, controlling transcription. Transcription factors ensure that a gene is used at the right time and that proteins are made in the right amounts. The complex structure of eukaryotic DNA also regulates transcription. Hox genes. They are responsible for the general body pattern of most animals. You can see how the different colors are responsible for different body patterns. Okay. RNA interference. this can stop the mRNA from translating its message. Almost, if you want to relate this to like an inhibitor with enzyme substrates, you can think of it like that conceptually, obviously not exactly the same. Mutations, a permanent change that occurs in a cell's DNA, is called a mutation. Three different types are point mutation, insertion, and deletion. So here you see, and I'll let you kind of dig into this on your own, but here you see the mutation type. And here's the normal sentence. Think of it just the big fat cat ate the wet rat, okay? And with these different mutations, right, notice they're 3 letters each. So they would be an example of each word in this case. Sorry, up here. Each word would be a codon. And so if a mutation occurs, and for example, this one, there is a mutation here where it affects only one codon. The codon before it is, quote, normal, and the codons after are normal, all right? But if you look at different types of mutations, one small mutation can actually change the whole sentence. Things like down here, okay? So anyway, I'll let you look into those. You can pause it, obviously. And then examples of diseases or disorders that come from these types of mutations. All right. Protein folding and stability. Substitutions also can lead to genetic disorders, can change both the folding and stability of the protein. Causes of mutations. They can occur spontaneously, or what we think of as spontaneous, because, with technology and things like that, as technology advances, scientists, researchers can discover more and more. So it could be spontaneous, but it might not be. However, we just don't know yet as a scientific community, as a society. Anyway, moving on. Chemicals and radiation also can damage DNA. High energy forms of radiation, such as X rays or gamma rays, are highly mutagenic. Body cell versus sex cell mutations. Somatic cell mutations are not passed on to the next generation. However, mutations that occur in sex cells are passed on to the organism's offspring and will be present in every cell of the offspring. And if we kind of relate this slide and the last slide, we think of X-rays as being highly mutagenic. Well, in order to protect human sex cells, if anyone has ever gotten an X-ray, you hopefully were given a heavy vest to wear. That's a lead vest to protect your sex cells from the X-ray radiation so that this doesn't occur. And that's it for chapter 12. Let me know what you guys thought. Sorry for not being in presentation mode for the first section. I'm just trying to evolve and improve as time goes on. So I hope everyone's doing well. And have a good day. This is Mr. Case signing off.